Welcome to this explainer. We're jumping right into one of the most mind-bending frontiers in modern biology today. Seriously, think about your own body for a second. For decades, we kind of just accepted that aging and cellular development were a strict one-way street, right? You grow up, your cells get specialized, they age, and, well, eventually they stop working. But what if I told you that cellular identity is actually an actively maintained state, not a permanent destination? Like, what if we could actually hit rewind? Okay, let's dive into this. Back in 2006, a Japanese scientist named Shinya Yamanaka made a discovery that completely shattered our understanding of biology. It was a breakthrough so profound it won him the Nobel Prize just 6 years later. He found that the biological clock isn't just a countdown timer leading us to old age, it's actually a fully reversible switch. By introducing a very specific set of proteins into adult cells, he proved, without a doubt, that we can revert mature, specialized cells all the way back into the equivalent of embryonic stem cells. To really grasp the magnitude of this, you've got to understand how we used to look at things. Historically, cellular development was imagined as Conrad Hall Waddington's epigenetic landscape. Picture a marble rolling down a rugged hill. As it rolls down, it settles into these deep grooves, becoming a specialized skin cell, a neuron, or a heart cell. And gravity only goes one way, right? But Yamanaka's reprogramming completely changes the laws of biological physics. His discovery acts like a molecular motor that is actively pushing that marble backward, defying gravity, and restoring the cell's ability to become absolutely anything. Here is our roadmap for the explainer. We'll start with the blueprint of age reversal, trace the journey to pluripotency, talk about resetting the epigenetic landscape, look at some expert views on translation, and wrap up with the technical and safety hurdles. Starting with Section 1, the blueprint of age reversal, focusing on the Yamanaka factors. So the crucial point is that this massive biological transformation is driven by just 4 transcription factors. They're known as the Yamanaka factors, or OSKM for short. That's OCT4, SOX2, KLF4, and c-MYC. You can really think of these 4 proteins as a molecular eraser. Imagine your DNA is like the hardware of a computer, but epigenetics are the software, the chemical sticky notes that tell a cell to act like a skin cell or a bone cell. The OSKM factors systematically wipe away those sticky notes, turning off adult genes and rebooting the cell straight back to its factory settings. That's a state of limitless potential called pluripotency. It's an incredibly elegant, systemic wipe of the cell's entire life history. Moving on to Section 2, the journey to pluripotency, a cellular rewind. Now, the journey from a specialized somatic cell to an induced pluripotent stem cell, an iPSC, doesn't happen overnight. It takes weeks, and it unfolds in 3 very distinct phases. Step 1 is initiation. Within hours, the cell undergoes a massive epigenetic awakening. It literally sheds its adult identity and changes how it eats, shifting its metabolism to glycolysis. It's gulping down energy the exact same way a rapidly dividing embryo does. Step 2, maturation, is basically pure chaos. It's highly stochastic, meaning genes are just firing randomly and unpredictably. Actually, a lot of cells fail and die here because the biological stress is just so intense. But if a cell survives the chaos, it hits Step 3, stabilization. Here, key master genes, like the SOX2 locus, slam into gear, strictly locking the cell into a stable, fully reset, pluripotent state. It's an incredible biological gauntlet. All right, Section 3, resetting the epigenetic landscape. Let's compare full versus partial reprogramming. And this brilliantly illustrates a massive strategic divide in the field right now. Full reprogramming takes the cell all the way back to an embryo-like state. Sure, it provides basically 100% rejuvenation, but the cell completely forgets what it is. If we do this inside your body, it's incredibly dangerous. You definitely don't want your heart cells suddenly forgetting they need to pump blood. So partial reprogramming is the safer, more subtle alternative. By applying the OSKM factors for just a short burst, we strip away the aging markers, but stop the process before the cell loses its identity. It stays a heart cell or a neuron. Is just a biologically younger, healthier version of one. It's definitely safer, but achieving that precise control on the molecular level, immensely complex. Section 4, expert views on translation. Let's talk about taking this from the lab to the clinic. Joe Betts-LaCroix, the CEO of Retro Biosciences, points out a really tough reality. Regulators just don't treat aging as a disease yet. So their strategy is to use stepping stone indications. Retro is focusing on full reprogramming outside the body, They take diseased cells out, like bone marrow, or reprogram them to a fully youthful state, carefully control their quality, and then put them back into the patient as a replacement therapy. So they aren't chasing a magic immortality pill right away. They are solving specific, defined medical problems to build a strong foundation of safety. Meanwhile, Sharon Rosenzweig-Lipson at Life Biosciences is leading the charge on partial reprogramming in vivo, meaning inside the living body. And this is wild. They have already successfully used 3 of the factors, OSK, to reverse aging in the retina and actually restore vision in animal models of glaucoma. By targeting the eye, which is a nicely contained organ, they can safely demonstrate that partial reprogramming restores youthful function without making the eye cells lose their identity. It's a localized, contained proof of concept, but it has absolutely massive implications for future therapies. Taking a different organ-by-organ approach, Yuri Daigan of Youth Bio targets the brain, specifically aiming at Alzheimer's disease. Now, the brain is incredibly appealing from a safety standpoint. Because neurons rarely divide, they are stubbornly locked into their identities, unlike, say, liver cells, which are constantly regenerating. This makes the brain uniquely resistant to going haywire during treatment. Delivering the therapy locally to the brain maximizes the rejuvenation benefit while really minimizing the risk to the rest of the body. Summarizing the incredible care required here, Vittorio Sebastiano of Turn Biotechnologies emphasizes that safety is the ultimate design constraint. He strongly argues against blunt, permanent genetic resets. Instead, he advocates for delivering these reprogramming factors as temporary mRNAs. Think of it kind of like a self-destructing Snapchat message. The instructions enter the cell, trigger the rejuvenation, and then just vanish without permanently splicing into your DNA. It allows for transient, perfectly tunable intervention. Let's move to and see how this builds into Section 5, Technical and Safety Hurdles. Time for the reality check. Okay, we have to talk about the realities here, starting with the darkest shadow hanging over this field, the teratoma. Because fully reprogrammed pluripotent cells can become absolutely anything. Even a tiny mistake can spawn a chaotic tumor growing totally random tissue types. We're talking literal teeth, hair, and bone growing where they absolutely shouldn't be. No way around it, it's a bizarre, terrifying risk. And it's exactly why biotech companies must develop ultra-sensitive purification methods and even engineer genetic suicide switches into the therapies. If the cells grow out of control, you need a safe way to instantly kill them. Then we have the massive challenge of delivery. I mean, how do you actually get the Yamanaka factors into the right cells safely? Traditionally, scientists used viral vectors. Viruses are super great at entering cells, but they carry the risk of insertional mutagenesis. That's essentially accidentally cutting the wrong wire in the human genome and causing cancer. Yikes. That's why the field is pivoting hard towards safer non-viral methods like mRNA and lipid nanoparticles. These modern methods deliver the reprogramming instructions temporarily without permanently altering your genome, and they largely avoid triggering massive immune responses. Huge upgrade. And look, even if the biology works perfectly and the delivery is completely safe, we still have to manufacture this stuff. Scaling this cure is a monumental barrier. We aren't just brewing standard chemicals here. We are babysitting living, highly volatile cells. Reprogramming cells is an incredibly inefficient process right now. Doing this safely for humans requires maintaining absolute sterility and battling high production costs in multi-million-dollar GMP facilities. Ensuring that every single batch of cells is identical and perfectly safe requires infrastructure that, frankly, is incredibly difficult and wildly expensive to scale globally. Which leaves us with this final thought. We now know for a fact that the biological clock is not fixed. We possess the molecular tools to push the marble of cellular identity right back up the Waddington Hill. But as this unbelievable science transitions from petri dishes to actual human patients, will our precision engineering and manufacturing capabilities conquer the very real risks of tumor genesis and instability? The answer to that question will literally determine whether cellular reprogramming becomes the ultimate medical revolution of our lifetime. Thanks for joining me on this explainer. Keep asking those big questions, and I'll catch you next time.